Welcome everyone to this second webinar of the International Eosinophil Society on eosinophils and cancer. Welcome to those who already joined us for the January webinar on eosinophils and COVID-19, and welcome to newcomers. For those of you who don't know of our society, I encourage you to visit our website to see what we do, who we are, and to eventually consider joining us as members. Today's session will focus on eosinophils and cancer. Many of you know that there's a wealth of clinical and experimental data indicating that the role played by eosinophils in cancer are varied. Blood eosinophils may be associated with favorable or unfavorable outcomes, depending namely on the type of cancer and the timing of blood counts with regard to the initiation of treatment. Eosinophils are also known to be present in the tumor itself. They're called tumor infiltrating eosinophils, where conflicting data show they may display pro-tumoral properties or anti-tumoral functions. In addition to the functions of tumor cells themselves, eosinophil heterogeneity and the complexity of local interactions with other immune cells in the tumor microenvironment likely account for this variability in experimental findings. This is a major focus of current research on eosinophils and cancer. So today we're going to have some fascinating presentations on these different topics. We're going to hear how eosinophils and their interactions with cells that are present in the tumor microenvironment contribute to anti-tumor growth. And we will hear how eosinophils are involved in the anti-tumor response that is boosted by immune checkpoint blockade. Before leaving you in the very competent hands of our moderator, I would like to highlight a few technical aspects of today's meeting. The presentations will be made live and they will last 20 minutes. And there will be 5 minutes for discussion directly after each presentation. The speakers will answer questions that you have entered in the chat box in the question and answer section, which is on the right-hand side of your slides. And our moderator today, who is Amy Klion from the National Institute of Health, will voice these questions on your behalf. So please do not hesitate to enter your questions into the question and answer boxes during the presentations themselves so that she can already select and prepare the questions. The presentations will be recorded and they will be uploaded to our website within days after today's webinar. And after the webinar, you will receive a survey and we really ask and encourage you to answer that survey so that we can further improve our competencies in organizing future webinars. And with that, I will leave you with our moderator.
So, hello, good morning, good evening, or good night, depending on where you are. It's my great pleasure to introduce 3 outstanding speakers. Their biographies can be found on the website of the International Eosinophil Society. I'm sure you'd much rather hear from them than from me, so I'm going to just introduce them very briefly. Professor Gabrielle Belz is our first speaker. She's the laboratory head at the Walter and Eliza Hall Institute of Medical Research in Australia and is joining us very late at night. She will be talking to us about divergent pathways in innate lymphoid cells that reshape melanoma outcomes and bring eosinophils into the game. So welcome, Dr. Belz, and thank you for being with us.
So thank you very much. I hope you can see that well. So today I wanted to share a story that we've been developing actually over the last 4 years, trying to understand how initially innate lymphoid cells contributed to melanoma outcomes, but that really brought us into a space that we weren't expert in and that Ariel has helped us with, and that's understanding how eosinophils contribute to this sort of protection. So melanoma is a deadly disease. Worldwide, America and Australia have the highest number and frequency of deaths. And for us, it's because of the location in Australia and exposure to sun. And depending on at what stage you're actually diagnosed with melanoma, determines the outcome for your disease. So the overall survival is about 50%. And this is despite the enormous progress we've made in immunotherapies. So in my opinion, 50% is not really great odds. And so we're really interested in how can we improve the treatments that we give to patients, and how can we identify perhaps biomarkers that allow us to understand and identify our disease at a stage where we can treat it much more efficiently. So my lab spends most of its time looking at innate lymphoid cells, and these are really fascinating cells. They're divided into a number of populations that really parallel CD4 T cells. So there are three groups. So group 1, or ILC1s, which are characterized by the expression of the transcription factor TBET. And they compose NK cells and ILC1s that are really known for their role in immunosurveillance. And then there's a group 2 cells, so ILC2s that produce the cytokines IL-5 and IL-13. And they're often found in allergic diseases or in responses to parasites. And then ILC3s, which we've actually spent a lot of our time trying to understand what ILC3s do, mainly because these are involved in responses to microbial infections, but also they're the building blocks of how lymph nodes and secondary lymphoid tissues are actually formed. And so I was joined in the lab a few years ago by a very talented postdoc, Nicholas Jacquelot, and he's extraordinarily interested in melanoma and very passionate about trying to understand how ILCs might contribute to melanoma tumours. And so we examined this in a model in which, so this is a model of a mouse where BRAF can cooperate with P10, and that we can inducibly cause this deletion of P10 to actually induce spontaneous models of melanoma. So these mimic what happens in humans a lot more closely than some of the adoptive transfer of tumour cells does. And so we've used this model to validate how close this might represent to human melanomas. And so you can just see in these slides the development of these tumours. So after about 20 days, we see that spontaneous intradermal tumours are forming. And so the first thing we did was ask a very, very simple question, and that is, are NK cells actually, or NK cells and ILCs and other ILCs actually recruited into tumours? So do they play any role whatsoever? And so I'm just showing you the gating strategy here. And so ILCs, they're essentially described as a lineage negative population. They do express a lot of the different markers seen in conventional T cells, but they don't express CD3 and they don't express CD19. And we use a whole host of markers to exclude conventional populations. But then we use CD49 as a marker of ILC1s. So they're EMAS positive, CD49 positive, CD49A positive. And then NK cells are NKp46 positive and CD49B positive. From there, we're able to define ILC2s by their expression of GATA3 and ILC3s by their expression of raw gamma T. And so you can see if we enumerate these cells that are infiltrating these tumours, you can see that there's quite a lot of these cells, relatively small numbers, but that all these populations are quite well represented. And that we found that, in fact, one population, the ILC2s, were represented at a somewhat higher rate. So this is really interesting, but we wanted to know, does this happen in humans? And so we collaborated with some people at the Peter MacCallum Institute, which is just down the road from the WEHI. And they were doing some analyses of human tissues, and they really weren't interested in ILCs at all. They were looking at T cells. And so we actually were able to take advantage of all the data that they had shelved. They very fortunately collected all their data on single cell sequencing and all their immunohistochemistry. And we were able to use all the stuff that they weren't using. And so what you can see here is these very small numbers of ILC2s that exist in the tissues. And so we can only stain for a small number of transcription factors in these tissues or markers, but we can use a selective gating system using GATA3 to identify what are these ILC2s. So do the ILCs actually play a role in the melanoma response? And we have combined our analyses of human tissues with those of mouse tissues so that we can really distinctly dissect the different pathways involved. And so here you can see B6 mice, so fully immunocompetent mice, RAG1 deficient mice that lack the adaptive immune system, so no T and B cells, and then RAG2 gamma C mice that lack ILCs, T cells, and B cells. And if we now challenge those mice with a RET tumour, you can see that if you lack the ILCs, you have a much poorer response in the tumor. And so this shows you the contribution of the adaptive immune system, and then subtracting from that the ILCs. So this is a really staggered response. And so you're most likely to live if you have all of your immune cells, and you have a very poor response if you don't have your ILCs. So then that really challenged us to ask which ILCs. And so there's this model where you can cross an IL-7 receptor Cree mouse with raw alpha flox mice, and this was developed by Andrew Mackenzie, who we collaborate with in the UK. And this really fairly selectively deletes out the ILC2s. And we can challenge those mice, again with the ret tumor. On this left-hand side, you can just see what the depletion looks like for the small intestine and for the tumor microenvironment. And we have this very selective, substantial reduction of the ILC2s that exist in these models. So in the small intestine, we lose most of our ILC2s, and there's virtually nothing in the tumor. And if we look at the outcome for those mice, you can see if you don't have those ILC2s, you have much larger tumors, and you end up having a much poorer outcome for your responses. So how do we work out exactly what the different ILCs are doing? We've used this model that a number of labs who work on ILCs have used. And so this is a model that I'm always really super surprised that it actually works, because I think it's a real challenge for the cells that we transfer into RAG2 gamma Cs. So here we selectively sort out these different populations of cells. So the CLPs that can generate all the different types of immune cells, the ALPs that can generate all the ILCs, and then the various ILC progenitors. We adoptively transfer about 5,000 cells into our RAG2 gamma C mice, and then give them about 4 weeks to reconstitute, and then we can challenge them with the tumor. And if we do that, we see that if we have the RAG mice, they are intermediate. So again, the similar data to what we saw before. If you have B and T cells, they do quite well. But if you don't have your ILCs, you have a much poorer outcome. And if you take the adoptive transfer system, where we transfer in now the CLPs, ALPs, all the ILC2 progenitors, really strikingly, you can see this situation where if you just have the ILC2 progenitors that give rise only to ILC2s in these mice, it reconstitutes most of the protective effect against the tumor. So what does this mean in terms of humans? And so we work with bioinformatician Melissa Davis, who we've been able to develop signatures of these different cell types, and then she's been able to use machine learning with her postdoc, Sarul, who does a fantastic job of really trying to dissect out in these databases of melanoma whether we have effects due to the different cell types that we've identified. And so this just shows you, if you have a high number of ILC2s, so as per a signature, you have a much better outcome for the prognosis. So what are the molecular pathways involved in this ILC-dependent anti-tumor immunity? Well, this becomes a bit of a black box. So we resorted to doing some single-cell RNA sequencing. And so we sequenced all the cells that were in three different tumors from our mice. And you can see here's our ILC2. And so it's just a very small number of cells, but a quite discrete gathering of those cells in the tumor. So if we hone down on what those cells are doing, so here's your ILC2s here, and this is just a selection of genes that, that are driving this response. And so what you can see that there's, classically the IL-13 is being produced, but what we were really interested in is you could see amongst these different cell types that the ILC2s were producing GM-CSF. And we were really lucky at the time that at WeHi, Ian Wicks was generating with Warren Alexander a GM-CSF deleted mouse. And so we were able to take advantage of that mouse. But first up, we looked to see which cells are actually producing GM-CSF. And so this is by intracellular staining. And quite paradoxically, you can see the NK cells, which can produce GM-CSF, are actually not doing this in the tumor microenvironment. We're able to use mice that are selectively depleted of NK cells to show that in the context of this tumor, the NK cells are not actually having an effect. So they have a profound effect on the metastasis of the tumors, but not in this initial establishment and escape of a tumor cell. And in this case, virtually all the cells in the tumor that are producing GM-CSF are the ILC2s. So what's GM-CSF doing? Well, if we take a knockout mouse, you can see that if you don't have GM-CSF, you actually have a much worse prognosis and much larger tumors. And if we then go back to the TCGA database and do some machine learning around gene signatures to do with GM-CSF, we now find that if you don't have GM-CSF in your tumor in a human, you also do much worse over time. And so these ILC2s are driving the formation of the development of GM-CSF in the local anti-tumor response, but how does this actually work? And so this is where we started to become drawn into the eosinophil world. And so if we look at the different types of cells that are being generated and recruited into the tumor, and this is in the adoptive transfer system. So now we've got sort of a complicated system where the RAG2 gamma Cs are being reconstituted with these different ILC progenitors. And so if we just focus on this yellow bar, you can see that this is sufficient to recruit significantly high numbers of eosinophils. And so we thought the eosinophils therefore must be playing an active role, because what we know is these eosinophils are actually being driven by the IL-5 produced by the ILC2s. And so you can see here we replicate this process. So if we delete the ILC2s, you actually get a reduced number of eosinophils into the tumor. So there's a relationship between these different types of cells. So if we look at eosinophil deficient mice, so these are the film mice, again, you see that lack of eosinophils drives increased tumor size and a much poorer prognosis. And we went back to the TCGA database, and now we're modeling ILC2s, high levels of ILC2s with high levels of eosinophils. And so we do all the different groups that might be driving that or playing a role in it. And what you can see is if you have high levels of ILC2s and high levels of eosinophils, you've got a much better prognosis. And so we were really stuck for a while as what the mechanism might be for this. And so we had been working on a separate project. We were looking at PD-1. And here's the single cell data that just shows you the expression of PD-1. And you can see here, the ILC2s are producing PD-1. So normally we think of this in the context of T cells. And this is just the antibody staining on that and the increased frequency of ILC2s that are expressing PD-1 compared to T cells. So it's sort of an equal number. And if we look in our sections in humans, we can see that these PD-1 positive ILC2s are now near our tumors. So in the PD-1 knockout mice, again, if you lack, or in this case, it's the reverse situation. If you lack PD-1, you actually have a better prognosis, which was a slight surprise to us. And this was also true if you adoptively transfer PD-1 deficient ILC2s into the mice. And so we wondered what the mechanism was there. And so we did what a number of groups are doing, and that is to stimulate the production of ILC2s with IL-33. And what this showed us is when you do this, you actually amplify the expression of PD-1. So you're driving a very forward response, but at the same time putting a brake on by the expression of the PD-1. And these data just show that. And so you can see here with the IL-33, you have this high expression of PD-1. And so while you're trying to drive this very positive response, you get this quite negative outcome. So the question was, could we combine the responses, block PD-1, but drive the amplification of ILC2s? And when we do this, you actually improve the prognosis, reduce the tumor sizes. And this is just shown here, these very small tumors now when you're blocking the response. And here you can see that the IL-33 is driving the production of eosinophils concurrently. And this is enumerated here. So the more IL-33 you have and the blocking of PD-1, you recruit many more of these eosinophils to produce a very productively outcome for the animals. So really to summarize what's going on here, what we think is that the ILC2s are coming into the tumor very early in the response, and that this is really critical for producing IL-5 and GM-CSF, and this is recruiting eosinophils. And it's actually inducing the effector functions of those eosinophils. And simultaneously, it's inducing the PD-1 expression on the ILC2s. And so now if we can come in and actually block that PD-1 expression, we can really facilitate this little circuit of ILC2s driving eosinophils to actually restrict the development of these tumours in the primary setting. So I'll leave it at that. It requires me to acknowledge a really large number of people who've been involved in this project. And I won't go through them all, but there's quite a number who are in Melbourne. But very fortunately, through this process of trying to work out what's been going on, we were able to collaborate with Ariel and Sharon in helping us understand what eosinophils are doing. And now we're really very, very super interested in trying to understand how essentially these innate networks are driving or setting up this context in which tumours are limited in their progression, and how we might take advantage of that setup to actually stop tumours from progressing to that metastatic form, which are very, very difficult to handle in the context of a clinical setting. So thank you very much.
So thank you very much for that thought-provoking talk. I have a question, but I'm going to first go to the question and answers and let other people go first. So there's a question from Christophe Desmet. Have you compared ILC2s in nevi versus melanoma? Because that might hint at the progression of nevi towards melanoma.
Yeah, that's a really great question. So what we're setting up now with people at the Diamantina Institute have access to a library of about 300 different samples that they've been collecting historically and actually collecting forward. It gives us the opportunity using nanostring technology and the machine learning that we've developed with Melissa around that. So developing signatures that allow us to actually hone down in a single cell manner as to which cells are interacting with which. So this gives us the opportunity now, I mean, this is where technology has come into the fore with COVID, to be able to take advantage of all those samples that have been collected over time from humans to actually work out what is the temporospatial architecture and who's driving whom in this response. And ideally, people really ignore eosinophils in the path lab to be able to say, well, if you could do some sort of differential around eosinophils, which are highly recognizable, versus ILCs, which are unrecognizable because they're far too infrequent, you could come up with a diagnostic or prognostic test to stratify people into clinical treatments. And so that's our sort of long game on that.
So that kind of segues into my question, which was about the timing. So there's a lot of data from allergic inflammation about ILC2s driving eosinophils, but eosinophils can directly respond to IL-33. They have the ST2 receptor. And one of the problems that we see in a lot of the work that's both published and in our lab is that you don't get an RNA signature from eosinophils when you do single cells. So they're completely missing from the plot, but it doesn't mean that they're not there. So do you have any idea what the timing is and how much of this is ILCs driving eosinophils and how much of it might be eosinophils themselves responding to to tissue destruction and damps and things of that nature?
Yeah, so the way in which we've looked at this problem is really the ILCs driving the eosinophils. But after a lot of discussions that we've had around this, there's clearly a lot to be done in what the eosinophils are actually driving, what the feedback loops are. And so that's what we're really interested in, working out ways of doing that now, because I don't think it's a one-way circuit. And this circuit gets disarmed like T cells do. And if we can understand both arms contributing the eosinophil and the ILC2 circuit, we can probably create that. So this is where the paradox around ILC2s comes, is that as they become disarmed, they become destructive. But if we can understand that circuit and stop the dysregulation, so this is where the blocking of PD-1 comes in, we can prolong that protective arm through the eosinophil and their direct response on the tumour cells. So there's still a lot to be done in that. And yeah, obviously there's some technical issues around that. And that's where collaboration with Ariel and Sharon is so critically important, because you guys are the experts in that space. And so that's where we can probably help each other in really starting to understand this very early response that can set the tumor up to not escape, basically.
So Christophe Desmet had a follow-up question about your data on the GM-CSF that kind of links into that a little bit. So he said that GM-CSF is very reminiscent of the GM-CSF licensing of anti-tumoral activity in intestinal eosinophils. Do you expect there's a direct role of GM-CSF on eosinophil anti-tumor function in melanoma?
Yeah, that's a good question. We've done a few experiments where we've co-cultured the eosinophils with GM-CSF. And so we do induce some of the sort of effective functions, but it's not inducing actually lytic functions from them. But again, that doesn't have to be the way in which that is mediated. We often think about that for T cells and NK cells. But I think it's a bit more nuanced than that. And so we have to do more work moving forward with that.
Mats Johansson asks, is there a risk now with the new anti-eosinophil therapies, anti-IL-5 or anti-IL-33 biologics, that they may increase risk for melanoma?
Yeah, that's a great question. I think there are things that we have to look at and consider because I think we often take very blunt-ended approaches to these things. But with you know, the connections through clinics, it'll be possible to integrate some of those samples and data sets and clinical parameters to now sort of consider those in the context of the outcomes for those patients.
Thank you. Mark Rothenberg asks, eosinophils are not classically part of the melanoma pathology. How variable and many eosinophils are infiltrating human melanoma? And have you transferred GM-CSF knockout ILC2 to see if they have a defect in eliciting the anti-melanoma effect?
Yeah, so that's an experiment we've just done. And if you do transfer in GM-CSF knockout ILC2s, then you fail to recruit your eosinophils into the response. And we don't exactly know how that little network is occurring because the classical recruitment is through the IL-5. So there's still more work to be done around that, but clearly that GM-CSF is really important for that recruitment. Yeah, it's true. Depending on when you look at the melanoma, you do or you don't see eosinophils. And so that's one thing that we're trying to do is really quantitate the eosinophils and how we can use that as a parameter moving forward.
I know that you can see the Q&A and you can perhaps respond in more detail to Henry Stevenson Perez later. But what the end of his question is, what carcinogenesis model are you using? I think that should be answerable.
Right, so we're actually using mainly this spontaneous model. And so we're just doing the deletion with tamoxifen. And yeah, we're trying to use intradermal models that really try to mimic what's happening in humans as opposed to some of the other models that are available.
And finally, in the 1 minute that's left, Steve Ackerman asks, eosinophils express GM-CSF. Is some of the GM-CSF coming possibly from the eosinophils and driving their survival in the tumors?
Yeah, that's a possibility. So as I said, we've really looked from the point of view of the ILC2s. So the adoptive transfer of the eosinophils, you know, comes with technical challenges, which is why we've used the knockout animal. But you know, having the flox mouse, you know, will allow us to do some other experiments around that moving forward to try and understand what the eosinophil arm is actually doing without some of those complications. But I think, yeah, it's got to drive that. Somehow the survival of the ILC2s and the eosinophils has to be set up in order for that response to actually happen. And there's no reason to think that the eosinophils aren't driving a very substantial amount of that.
Thank you very much. We managed to get through all the questions in the Q&A box and in perfect time. So I think we'll move on to our second speaker. I'm waiting for the slide to come up. So our second speaker is Professor Ariel Munitz. He's the Associate Professor of the Department of Microbiology and Clinical Immunology at Sackler School of Medicine in Tel Aviv University. He's also on the executive board of the IES and has a long interest in eosinophils. And he's going to talk to us about metastasis-entrained eosinophils, enhancement of lymphocyte-mediated tumor suppression. Welcome, Ariel.
Thank you very much. I'll try to share my screen. Okay. So thank you very much for this invitation and really exciting seminar or webinar. And I'll be talking today a little bit about what we know about eosinophils and their interactions with lymphocytes. So basically, when we started to look into this topic, we started looking to the literature, and we came across this, I would say, seminal review article, which was published in the year 2000 by Hanahan and Weinberg. And if you only look at the citations, which is, according to this morning, 34,994 and probably still counting, you can see that the way that cancer was looked upon 20 years ago was with much of a focus on the cancer cell itself, on the mechanisms which drive the tumorigenic process within the tumor itself. However, 11 years after that, meaning 10 years ago, in a second review article, the same authors, which is now termed Hallmarks of Cancer, the Next Generation, they introduced also this concept of emerging hallmarks and emerging characteristics of the tumor microenvironment. And over there, you can see that tumor-promoting inflammation plays a key role. And in the contrast, avoiding immune destruction by tumor cells also plays a key role in the tumorigenic process. Now we're 10 years after this review, and today we know that the tumor microenvironment, and especially the immune system, they have critical roles in the tumorigenic process. And I think that this statement is best exemplified by the fact that immune checkpoint blockade has been recently been awarded the Nobel Prize. Hanjo and Allison got it for their seminal work on CTLA-4. But even with this, major breakthrough, there are still many patients who do not respond to therapy. And therefore, there is still an urgent need, which exists, to define the contribution of additional immune cells in the tumor microenvironment. And with that in mind, and with our ongoing interest in eosinophils, we started to look into the role of eosinophils, which, as we all know, have been traditionally studied in context of allergy and parasite infections. But if you really look into the literature over the past couple of years, you can see that eosinophils have been attributed with additional homeostatic functions which haven't been traditionally associated with these cells. For example, roles for eosinophils in obesity and metabolism, roles for eosinophils in muscle regeneration and liver regeneration, new roles for eosinophils even in innate immunity and adaptive immunity, specifically with synthesis of IgA and bacterial clearance in the GI tract. So what we had in mind is, as Florence suggested at the beginning, eosinophils have been suggested to participate in tumor immunology. And we know that they may have pleiotropic functions, either favorable or detrimental. And we were interested to start and understand what these cells do in the tumor microenvironment. And that was actually our first question. Which tumors are actually infiltrated by eosinophils? I mean, there are so many tumors, so which one should we focus on? And in order to answer this question, we actually decided to take an unbiased approach. So what we did is we took a bioinformatic analysis of 10,469 genomically profiled primary tumors. And these are taken from 22 different and distinct tumors. And by using a cell signature called the LM22 cell signature, which is deconvoluted by the CyberSort algorithm, we could clearly see that tumors are divided into three types in terms of their eosinophilic infiltration. There are tumors with high eosinophilic infiltration, tumors with intermediate, and obviously tumors with low eosinophilic infiltration. And what we wanted to do next is we wanted to validate these findings. So we gathered a cohort of about 2,900 patients from 6 different tumors, which are supposed to represent the different tumors which we analyze bioinformatically, meaning the low, intermediate, and high tumors. And we have clinical data for these relevant tumors, so we can actually start playing around with associations. And what we basically did is we stained all of these slides with anti-EPX antibodies, and this is a small picture, so you can't actually see the staining, but you will have to take a close look to see that these round black circles are actually eosinophils. And then we can screen these slides, and quantify them. And what we could see right away is that eosinophils, not surprisingly, will infiltrate into mucosal tumors. So our next goal was to find models which are mucosal models or mucosal tumor models where we think we could study eosinophils better. And one of the reasons we think that these models are better to study eosinophils because we all know that mucosal tissues are primary infiltrating sites at baseline for eosinophils. So you probably have a resident eosinophil population to start with, and not only eosinophils which are recruited to the tumor microenvironment. And one of the models which we started to study, and this was driven by Sharon Grisaro, a talented graduate student in my laboratory, she decided that she wants to focus on breast cancer. And breast cancer is the most common malignant disease in Western women. And in these patients, it's not the primary tumor which is driving the tumor-associated death, but it is actually the distant metastases which are the main cause of these deaths, of the death. And once these tumor cells are disseminated, they form carcinomas. They form carcinomas in various organs. And one of the most common organs which these cells disseminate and seed into are the lungs. And the lungs is obviously our mucosal tissue, and they're a natural habitat for eosinophils. Eosinophils know how to infiltrate into the lungs. And therefore, we thought that breast cancer-associated lung metastasis might provide us with a good model. So we next aim to define whether there are any eosinophils in human tissues of breast cancer-associated lung metastasis. So going through the database of the Soraski Medical Center, which is right next door to us, it took us a long time to identify 22 different patients, which we do have lung biopsies from their metastatic disease, and we stained them again with EPX. And as you can see here in this picture, there's a wide variety of eosinophil or EPX staining in these patients. On the left, you will see a patient which the eosinophil is seen intact and round, and on the right, patient 18848, where you see a large deposition of EPX, indicating that these eosinophils might be actually degranulating. And when we quantify whether these tumors or metastatic lungs are having or displaying degranulating eosinophils, you could see that about 41% of these tumors display degranulating eosinophils. So this kind of caught our attention, and we were further interested to understand this on a more mechanistic basis. And we had three major motivating questions. Our first motivating question was, how do eosinophils accumulate in this tumor microenvironment? And our second motivating question was, what is the function of these eosinophils in the tumor microenvironment? And the third question is, what is the mechanism in which eosinophils affect the tumorigenic process, if at all they do? And we took a rather simple model of tail vein injection of breast cancer tumor cells. And once you do that, we do a kinetic analysis of the eosinophil appearance in the lungs. And when we stain again for anti-EPX staining in the lungs, you could see again distinct locations for eosinophils in this metastatic lung. You can see that this is the tumor node over here. You can see a clear staining of intratumoral eosinophils. And you can also see eosinophils outside of the tumor area, which do not infiltrate the tumor. And when we quantify on a kinetic basis the appearance of eosinophils in two distinct models, the PYMT model, which is on the B6 background, or the 4T1 model, which is on the BALB-C background, you can see that, again, eosinophils display rather similar kinetics in both models, and they do infiltrate the lungs in both cases. Now, taking into account that in 2016, Mesnil et al. Published that, at least in the asthmatic lungs, there are two distinct eosinophil populations which can be identified by cyclic F high and low, and the expression of CD101. We wanted to assess whether the metastatic lung also expresses these two distinct populations. So basically, we took the same staining and the same protocol which they did, and we could clearly see that both in the PYMT injected mice and the 4T1 injected mice, you can see a resident population, which is here in gate R1, and the distinct, I would say, recruited population, which is in R2. And furthermore, when we gate on these populations of R1 and R2, looking into CD101, we could see that similar to what was found in asthmatic models, you can see that these eosinophils also have increased expression of CD101. We could not find any expression of CD62L in any of the subsets, in contrast to what was found in that paper, but we could also not find it in asthma as well. So we were asking ourselves, are eosinophils actually recruited to the tumor microenvironment? And the obvious question the obvious answer, sorry, is yes, because we could not find any proliferation of progenitor cells. We could not find any increased number of progenitor cells in the lung. But we really wanted to nail this and to understand whether the tumor microenvironment can actively recruit eosinophils. So we undertook a model where we inject or adoptively transfer eosinophils into naive delta double gamma mice here on the left, or into delta double gamma mice which receive saline, or those which receive PYMT cells, indicating that these are having a metastatic disease. And as you can see that when delta double gamma mice have a metastatic disease ongoing, they will recruit much more eosinophils into the lungs. And quite interestingly, the eosinophils which will be recruited into these lungs will have increased expression of cyclic F. And this increased expression of cyclic F already occurs within the blood. So there's something systemic going on which drives increased cyclic F expression on these eosinophils. So we were next interested to understand which factors induce eosinophil migration to this tumor microenvironment. And the natural suspect was CCR3 and eotaxin pathway. So taking advantage of CCR3 deficient mice, which we obtained from Mark Rothenberg, we could clearly see that CCR3 does not play here any role because there was no difference in the recruitment of eosinophils between the wild type and the CCR3 deficient mice. So we asked ourselves, do eosinophils migrate in response to any tumor secreted factors? And the simple answer is yes. When we take an in vitro approach, we take PYMT cells, the cultured media, the conditioned media of these PYMT cells, and use a relatively easy eosinophil transmigration assay, you can see that eosinophils migrate towards these PYMT secreted factors in vitro. We could also see that this migration is dependent on pertussis toxin, because when we treat eosinophils with pertussis toxin, we get complete inhibition of this response. So this is a GPCR dependent process. We can get similar data also with human eosinophils migrating towards human breast cancer cell conditioned media as well. We could also get this going on in vivo when we take a tumorless model. So when we inject intraperitoneal injections of this PYMT culture media and then assess eosinophil migration into the peritoneal cavity, we can get substantially increased levels of eosinophils coming in. So this is interesting. Eosinophils come into the metastatic lung, but what do they actually do in this lung? What is the role of eosinophils in lung metastasis? One of the things that we can do with these PYMT cells is that we can inject them, in the PYMT cells, we have a luciferase vector, and therefore when we inject mice with luciferin, we can actually visualize and therefore quantify the size of the tumor. And as you can see here in this picture, the delta double gamma mice, which lack eosinophils, have significantly increased tumors, and we can quantify that only to show that eosinophils probably have anti-tumorigenic activities in this model. Taking an additional approach, when we take anti-cyclic F antibodies and we deplete eosinophils, we can recapitulate the same phenotype also in wild-type mice. So basically, we can identify anti-tumorigenic activities for eosinophils in the tumor microenvironment. We were next interested to understand whether eosinophils can shape additional traits of additional characteristics of the tumor microenvironment. And given the fact that there is, I think, new data emerging from various laboratories around the world about an eosinophil T-cell crosstalk, we decided to look into the migration of CD8 positive T cells. So we stained for CD8 positive T cells here in green. The M cherry are the tumor cells. And we could clearly see that in the absence of eosinophils and increased tumor burden, you can get substantially decreased influx of CD8 positive T cells. Quite interestingly, we can also get substantially decreased levels of CD4 positive T cells as well. So basically, eosinophils will regulate infiltration of T cells into the tumor microenvironment, at least in this model, and the absence of eosinophils and increased tumor-associated burden is also associated with decreased lymphocyte recruitment. So going now back to the humans, we collaborated with Gabrielle and Melissa Davis and all the fellows in WeHi. And what we wanted to assess is take a bioinformatic approach, looking into the question, trying to answer the question whether we will find any increased lymphocyte subsets or any cell subsets in correlation with eosinophils. So this is quite a hard graph to follow. So I'll try to draw your attention basically to the upper left graph over here. And basically what you can see, what's written down means that this is where you have low levels of eosinophils, and where it's written up, this means that you have a higher level of eosinophils. And the only cell type which was associated with increased levels of eosinophils were CD8 positive T cell, or more correctly, a signature for a bioinformatic signature for CD8 positive T cells, which are activated. So this prompted us to try and understand whether the anti-tumorigenic effect of eosinophils was dependent on adaptive immunity. So we took the RAG2 gamma C deficient mice, which Gabrielle mentioned before, and when we now give this anti-cyclic F antibody and deplete eosinophils, we get a complete normalization of the phenotype. So basically, in the absence of this adaptive immune response, or more specifically, probably T cells, eosinophils lose their anti-tumorigenic activity. So the anti-tumorigenic activity of eosinophils in the lung, at least in models of metastasis, is dependent on lymphocytes. But how does the tumor microenvironment shape these activities for eosinophils? I mean, does the tumor microenvironment instruct eosinophils to promote this anti-tumorigenic effect? And in order to answer this question, we decided to take an unbiased approach of RNA sequencing and proteomics. So basically what you're seeing here on the left is an RNA sequencing of naive eosinophils in red and inflammatory and resident eosinophils on the right. So basically these are the resident and inflammatory eosinophils from the metastatic environment. And we did the same thing in proteomics. So we sort the cells and we subject them to a proteomic analysis. And you can again see the naive eosinophils in red and the metastatic eosinophils in blue and green. And what you could clearly see from here, using this PCA plot, is that the major factor which is actually differentiating between the naive eosinophils and these eosinophils in metastasis is actually the tumor microenvironment. So there's no difference between the eosinophils, whether they're recruited or resident. They actually have the exact same signature. And I would like to demonstrate that here in this heat plot, where you plot for different cytokines. This is one of the heat plots which we generated. And you can clearly see here two things. The first thing is that there is a distinct signature between naive and metastatic eosinophils. And the second is that there's a lot of heterogeneity going on in these eosinophils in lung metastasis. But basically, despite this heterogeneity, or maybe because of this heterogeneity, there's no difference between the inflammatory eosinophils or recruited eosinophils and the resident eosinophils. So we decided to call these eosinophils metastasis-entrained eosinophils because of the fact that the microenvironment induced such a robust signature in these cells. And in trying to assess what are the upstream regulators of these eosinophils, we went into some biological pathway analysis, and we could clearly see that these eosinophils respond nicely to cytokine stimuli, but not any cytokines, specifically interferon gamma and tumor necrosis factor. And in fact, when we use an IPA, ingenuity pathway analysis, we could see that the top upstream regulators are TNF alpha and interferon gamma. And in fact, STAT1, which is a hallmark adapter and signaling protein for interferon gamma signaling, was one of the top upstream regulators of eosinophils as well. And quite interestingly, when we do the same analysis on the proteomic approach, which we have, we also get here, and I'll draw your attention to this left corner, upper left corner. You can see that again, the response to interferon gamma is one of these hallmark characteristics of these metastasis-entrained eosinophils. So basically, this interferon gamma TNF alpha associated signature is really nicely described these eosinophils. So what we did next is we activated these eosinophils with TNF alpha and interferon gamma, and we assessed more than 100 cytokines or chemokines, and we could see a very nice signature for chemokines which can recruit T cells. So basically, we asked ourselves, can activated eosinophils suppress lung metastasis? And the answer is yes. When we adaptively transfer activated eosinophils into the lungs, and you can see this in a macroscopic slide, but also in a microscope histologically, you can see that control mice, you can see very nicely the tumor nodules, but the interferon gamma TNF-alpha activated eosinophils have significantly decreased lung tumor burden. And this is also associated with a significantly increased infiltration of CD8 positive and CD4 positive T cells. So if to summarize, and I can see Amy already giving me a look, if to summarize, when we are talking about a metastatic tumor mass in the lungs, or more correctly, a breast cancer-driven metastatic tumor in the lung, we have a CCR3 independent pathway that recruits eosinophils to this tumor mass. Now, one thing that we didn't show here is that the source of interferon gamma is coming primarily from NK cells and CD4 positive T cells. We don't know yet what the source of TNF-alpha is, but these two cytokines work together to activate eosinophils to secrete chemokines and probably additional factors, which will bring in the adaptive T lymphocytes, CD8 positive T cells and CD4 positive T cells to induce their anti-tumor activity. So basically, these data highlight a key eosinophil T cell crosstalk, which has anti-tumorigenic activities in lung metastasis. I would like to just finish by acknowledging really many people who assisted us in this work. First of all, to Shalom Glisavo, all of the data which I presented here from the beginning, all of this is her data, and she's going to be graduating soon, but also all of our good and amazing collaborators around the world. And thank you.
So thanks, Ariel, for a very data-rich, exciting talk. Unfortunately, we're really over time, and I don't want to give short shrift to the third speaker, so I'm only going to allow you to answer one question from Bruce Bachner, which is, does Siglec F itself play any role in the biology you described? Have you tested Siglec F null mice?
So no, we haven't tested Siglec F null mice. It's probably on the list, and we have to do that as well. Yeah.
All right, well, thank you very much for a really interesting talk. If there's time at the end of the third speaker and people have questions for Ariel, please put them in the Q&A and we'll address them then. So our last speaker today is now blocked by things on my screen, is Professor Viktor Umansky, and he's going to be talking to us about eosinophils in melanoma therapy with immune checkpoint inhibitors, which hopefully will tie together some of the things that we've heard so far. So thanks very much.
So thank you very much for this nice invitation and introduction. So I'm going now to make quickly this to share with you and with laser pointed. So first of all, thank you very much indeed. I worked for a long time with T cells and myeloid-derived suppressor cells. And as you know, these cells are playing an extremely important role in the tumor progression or tumor rejection. So if we can imagine the tumor microenvironment as from the immunological point of view as the meeting of bad and good guys. So you see here on the left side, the cells which are acknowledged to be a bad guys, and on the right side, at least at the moment, good ones. So one example is, for example, with B cells, which we consider all the time as tumor promoting cells, but now there are some data showing about anti-tumor effect. And now eosinophils are coming in the game, as you have heard from the previous two brilliant talks. Actually, I will be focused more in the interaction of eosinophils in melanoma with T cells. And as you have heard from the talk of Ariel, that eosinophils can produce some being activated by TNF and interferon gamma can produce CXCL9, 10, and 11 that could attract Th1 cells, whereas being activated by TNF and IL-4, they can produce CXCL17 and CXCL22 and attract Th2 cells. Now, just 5 years ago, approximately, we were actually very much interested in the paper published by our colleagues from German Cancer Research Center in Heidelberg, Nature Immunology, and the quintessence was it's exactly what actually Ariel told us several minutes ago, that activated eosinophils, the only thing this paper was dealing with melanoma, which is our particular interest. Activated eosinophils modulated melanoma microenvironment and improved the infiltration of T cells into the tumor by the accretion of CXL9, 10, and 11, and enhancing thereby tumor rejection and survival. And this they showed on the mouse model. And since we are also working in the University Medical Center in Mannheim, close to Heidelberg, in melanoma department, so we decided to look how it happened in the human setting. So we have a collection of tumor samples, and we did tissue microarray analysis, making 2 independent measurements, and these 3 groups having one group from benign navy, then primary melanoma, which was then afterwards dissected in 2 subgroups, metastatic and non-metastatic primary melanoma, and then melanoma metastasis. And we made actually our staining with Siglec-8. Unfortunately, we were not able at that time to do the double staining. And then what we found actually that we see that really the infiltration of tumors, so-called non-metastatic melanoma. So again, this is a primary tumor, but we know that this patient, they don't have any metastasis at the time of taking this tumor sample. And then we see a clear infiltration of Siglec-8 positive cells in these samples in comparison, for example, to metastatic malignant melanoma. It means primary tumors, but from patients having no metastasis, having metastasis at the moment, and also regional metastasis and distant metastasis. So they were infiltrated with very few Siglec positive cells. Then the next question was just to see how would be the infiltration of CD8, CD3 positive T cells, and CD45 are O positive memory T cells in the same samples which we tested for Siglec-8 eosinophil staining. You know that the CD3 T cells and CD45 are O positive memory T cells are considered to be a good prognostic factor in melanoma, and we would like just to have a look how it really happened if T cells are going together, if they are associated with the accumulated eosinophils. And the answer is really yes. So we found exactly in the same samples non-metastatic malignant melanoma, again, it's primary tumors from the patient without any metastasis. We found an infiltration of both CD3 positive T cells and CD45 are O positive memory T cells. Looking then next, how this accumulation of Siglec-8 positive eosinophils correlate with the progression-free survival. So we found that really Siglec-8 high samples were corresponding to the patient, showing the higher progression-free survival. And this was in both cases. In this case, we divide on numbers of Siglec-8 high or low in intralesional samples and also parts and perilesional parts. And in both cases, we saw the very highly statistically significant correlation. We looked also intralesional and perilesion. So on the overall survival, we found here also very strong tendency for the correlation of high infiltration with eosinophils with increased survival of these patients. Now, next step was to look, because as Ariel told, and we know that was a time very, very good for melanoma patients and also doctors and scientists with the establishment and introduction in the clinical practice of immune checkpoint inhibitors. So we started already like 7 years ago, looking when only ipilimumab was actually approved for use, to study the role of myeloid-derived suppressor cells at the time. And then in parallel, we looked also on the eosinophils. And we found, using a very simple clinical routine method of eosinophil counts, that in the responders to ipilimumab, there is a very quick and constant increase, significant increase in the numbers of eosinophils in the peripheral blood, whereas non-responders actually show no increase of eosinophil numbers. What is interesting that among the possible, we looked, of course, what could be the possible chemokines involved in the in this effect, and we found, unfortunately, there were not so many patients so to look to study, and we found that actually non-responder there is a decrease with the time concentration of one of the important chemokine attracting eosinophils, eotoxin 1 or CCL11. In the next study, which was done recently, when already anti-PD-1 antibodies were approved for the clinical application, like pembrolizumab or nivolumab, which was used in this study in combination with ipilimumab. So these were 32 patients which we studied for the presence and infiltration accumulation of eosinophils. So most of the patients were of stage 4 and showing a very good response. In total, we have 12 responders and 17 non-responders, and 3 patients so showing mixed response. We started again with the routine clinical analysis and found that responders showed an increase in the eosinophil numbers and frequency among leukocytes already after the first injection of immune checkpoint inhibitors. And it's actually in both groups happened. So in pembrolizumab alone or nivolumab and ipilimumab combined treatment. What then, of course, important for us was to apply flow cytometry, and we use classical approach, looking on CD66B positive, CCR3 positive, Siglec-8 positive cells, and we found again a striking increase already after first injection of nivolumab or pembrolizumab, the increase of the amount of eosinophils among total granulocytes and responders, whereas non-responders, they don't show this effect. Moreover, actually all responders showed an increase in the amount of eosinophils, smaller or larger, whereas non-responders in only 50% showed an increase, and other 50% even decrease after the treatment. We looked also on the some chemokines and cytokines which could play a role in the function of eosinophils like IL-16 and CCL2, and found that there is a tendency, strong tendency of correlation between the frequency of eosinophils in the peripheral blood and the presence of IL-16 and CCL2 in the serum of this patient after the treatment. Looking on the survival, we found that we have the situation when high presence of IL-16 actually correlates with the better survival in both overall survival and progression-free survival. I want to point out also the fact that we have also the increase of CXCL9 concentration in the serum in circulation of responders after the treatment, after the first injection. Actually, it's an early response, I want to stress. And CXCL, we don't know exactly whether this source of CXCL9 was only eosinophils, probably not, but we think that eosinophils are contributing a lot to this point. And the last piece of investigation we did, we wanted just really in this situation in the treatment situation to see the accumulation of eosinophils and T cells, and to see the correlation between this accumulation. And what we found actually that we after the treatment, we see the amount of increase in the amount of activated eosinophils, and in responders we found also the at least a good tendency, the correlation between the amount of eosinophils, numbers of eosinophils per millimeter, and number of CD8 T cells. So this was made after the treatment and in responders. Now, just to go to the end of the talk, actually what we would like to summarize that eosinophils, as we have heard also from the presentation of Ariel and Gabrielle, so playing an extremely important role and probably before underestimated in the tumor microenvironment. Of course, we were talking mostly about the anti-tumor role of eosinophils. We believe that especially in melanoma situations, so they are playing an important role in attracting T cells. Would be, of course, extremely important. What we are planning to do also to look probably on the possible bad side of eosinophil, if they have this side, possible immunosuppressive effect. There are some publications about this. But the other side, if really eosinophils will be able to, and probably also direct anti-tumor effect of eosinophils, but we're seeing that if eosinophils will be able to attract lymphocytes and in particular T cells, so this will be, of course, a great achievement. And at the end, I would like just to acknowledge the people who, from my group, and especially Sonia Simon, who made it a part of her medical doctor work, and also to show you two places where we are working. One, and it's still a picture before Corona pandemic. So when you see beautiful building of the KFZ, and also in University Medical Center. And this is also, you can see also our international collaboration. And thank you very much for your attention.
So thank you, Dr. Umansky, for the third in a really wonderful set of talks. I actually have a question for you. I would remind people to put their questions in the Q&A, after which Dr. Rufos will make an announcement, and then we'll come back. There were some additional questions for Dr. Munitz that we'll answer at the end. My question to you is, in that graph that you showed where the eosinophils go up in all the patients that responded, but also went up in some of the patients who didn't respond. Do you know what the difference is between the ones who did and didn't have eosinophilia and the non-responders is? Because it must be something other than the eosinophils, or some difference in eosinophil activation.
Right. Well, unfortunately, we could not study really deeply these patients who, because since they did not have any response, so they were withdrawn from the treatment. And the only thing we know that, and this is what we showed in some other slide, that this patient, of course, they survive worst. The only thing we don't know exactly whether they survived badly just because they don't have any treatment, or just because of the absence of eosinophil accumulation. But this is, of course, would be one of the next point. And actually, what is also very interesting to us, and we try to figure out, is whether eosinophils could express, for example, PD-L1 and be like a brother of myeloid-derived suppressor cells in this case. We did not find any correlation with this, so that's why I did not show this data. But they do express PD-1. And this is exactly what we discussed in the previous talk about the PD-1 expression. I mean, the talk of Gabrielle, that it could be that PD-1 is really could be one of the factors just playing a role. It's one thing. Another thing, what I would like to comment, just having this opportunity, about GM-CSF. Because, well, since we are working for like almost 15 years in the area of myeloid-derived suppressor cells, and we know that GM-CSF is one of the greatest factor inducing generation of these highly immunosuppressive MDCs. So it means that we have a really not black and white situation. We have really very complicated system. And I think it's very much dependent on the study, on the stage of the tumor development. Thank you.
Thank you. So while we're waiting for more questions, I'm going to turn it over to Dr. Rufos, who's going to make an announcement.
Hello again, and thank you to the three speakers for a very outstanding webinar. Once again, we're very happy to be hosting these webinars, and this was really extremely interesting. All three topics very complementary. And I would just like to, before everyone signs off, to announce the program for the next meetings. Our next webinar is going to be held on Wednesday, March 24th, same time as today. So this is actually not the last Wednesday of the month of March, and that's an important exception to the rule that we had decided upon. Note your agendas. The topic is going to be regulatory eosinophils, and I think we heard some very interesting data today, namely by Ariel Munitz, suggesting some plasticity among eosinophil subsets. So I think it's going to be very, very interesting to hear these presentations on regulatory eosinophils, and we already have our full lineup. It's also, I think, going to be an outstanding webinar. We also have the webinars programmed for the months of April and May. The month of April is going to be a memorial to Barry Kay, and will be dedicated to airway eosinophilia. The May webinar is going to be presented by trainees. For the month of May, we are also planning to have an abstract submission deadline for an abstract, a series of abstracts that will be presented, selected and presented in September or October. So please also, all of you, mark your agendas for the abstract submission that should be sometime in May, and we'll probably be writing out emails very shortly to specify what the timelines are. We'd also like to just, no, I'm sorry, I think that's all I wanted to say actually, in terms of organization of the next meetings. So in general, the following meetings, with the exception of the one that will be held in March, are going to be the last Wednesdays of each month, same times today. So with that, I'll go back to the questions. I also want to thank Amy, obviously, for moderating this session. It was the timing is perfect. And and the questions as well. Thank you very much to everyone.
All right, well, thank you, Florence. Hopefully, both Dr. Umansky and Ariel, as well as many of the rest of you, can stay on. We have a couple of interesting questions left for both of our last two presenters. So there is a question from an anonymous attendee, reminiscent of a prior question about whether anti-IL-5 therapies or eosinophil depleting therapies could result in the increased prevalence of malignancies. And I'm going to take the liberty of saying there's no data to support that yet. And so I think a discussion of that is, we're starting to think about maybe that needs to be the topic of one of our future seminar series. I think it's a very interesting topic, but would require a very long answer for which there's no data. So if I can go back to some of the earlier questions. So Steve Ackerman asked Dr. Munitz, what GPCR and ligand are responsible for the recruitment of eosinophils in a CCR3 independent manner? Because that wasn't discussed. And if I can add something on for Dr. Umansky, you showed that there was a decrease in eotaxin 1. And so your data suggests maybe eotaxin is involved. Ariel's data suggested maybe eotaxin isn't involved. And so maybe Ariel can answer first, and then Dr. Umansky can add his comments about his presentation.
Yeah, so in terms of which GPCR signaling molecule is involved, we don't have a definite answer for that yet. But what we did is we took the secretome of these PYM T cells and we subjected it to mass spec. And then we analyzed it in terms of which GPCR signaling molecules are increased and which of them are known to induce granulocyte chemotaxis. And when we overlay between the two, we find 3 candidates. One of them is annexin A1, the second is CCL9, and the third is CX3CL1. Now, we started working with pharmacological inhibitors of annexin A1, and at least in vitro, they do suppress eosinophil migration towards PYM T cells. And this is interesting because this indicates that it might be a pathway which is mediated by FPR1, FPR2 receptors, which are also receptors of FNET-Lu-Phi. So we think that this might be one pathway. We weren't able yet to see this pathway operative in vivo. And in terms of CCR3, I would start, and probably Viktor could follow after I say what I say. I think it's really dependent on which tumor microenvironment the eosinophils are recruited to. I don't think that it's going to be a single molecule for a single tumor type, but Viktor.
Yeah, thank you, Ariel. You mean CCL11, yeah? So this is the eotoxin. Yes, of course. First of all, we need, of course, to collect much more data about this, and then please also don't forget that this is actually the situation in the circulation. So of course we have to. It's our next step to look deeper what happened in the tumor, right? Because of course, we're saying that it's very important to attract tumor-specific T cells into the circulation, but of course. Extremely important is also to attract them into the tumor. And so the next step will be just to find whether they're attractive or not. The point is that sometimes we don't have really the information after the treatment with ICI, so immune checkpoint inhibitors, from the point of tumor, because unfortunately, tumor is already removed in this case in melanoma patient, and they have mostly numerous but still small metastases which are not removable, and it's the problem. That's why a lot of people are proposing really to focus, especially thinking about possible markers which could correlate with prognosis, just to focus on to the circulation, to the peripheral blood, where we can have the approach practically every moment.
Okay, and then the last question from Mark Rothenberg to Ariel. Your model is proposing that eosinophils are middlemen rather than direct killers. Doesn't the degranulating eosinophil, like the ones behind me, suggest both roles? Yeah,
it's a good question, but I think that here we might be misled by earlier observations that eosinophil granule proteins can kill tumor cells in vitro, and it is not necessarily the case. When eosinophils degranulate, the granule proteins come out and are also accompanied by additional cytokines and chemokines, which are also present in these granules. So it is not only the effects of the possible cytotoxic effect of these cells. And I think that we have to really be careful about what's going on in vitro, where eosinophils can kill tumor cells in vitro, and whether this actually happens directly in vivo. But our data suggests that in this model, it's indeed indirect.
All right, well, thank you. Last question from Mario Lacouture. Adverse events from immunotherapies have been associated with eosinophils. Do you think they play a pathogenic role? And I'll assume this is for Professor Umansky, although it doesn't say.
Yeah, it's a great question, really, very interesting. And by the way, it's really exactly the point which we would like to study, because we collected now really a group of patients, so showing the adverse events. And of course, most of these adverse events are immune, so to say, mediated or dependent. And just to look whether actually the main aim is just myeloid-derived suppressor cells, whether it's somehow dealing with suppression or less suppression, it could be also interesting. But at this moment, we would like also to look on eosinophils. My prediction actually, but you know, you never know, but my just feeling would be that both cells probably could play a role. And depending again on the stage and the individual situation. So it means that it makes our immunomonitoring more complicated, right? That we have to look on also on this small population of eosinophils. Not only this great brother granulocytes, you know, like neutrophils, for example, or myeloid cells, monocytes. Sure.
Thank you very much, everybody, the speakers for really terrific talks, and all of the people who asked questions for such interesting questions. And hopefully we'll see you all next time. Florence, anything?
Thank you and goodbye, everyone. So please note your agendas for the next webinars. They're going to be as interesting as this one. And also you'll be receiving that evaluation form after this. Don't hesitate to make any comments or suggestions that you might find useful for further improvement. And finally, thank you. Have a nice day. And keep safe, everyone. Bye-bye.
Yeah.